complex generics, workshop with the characterization of complex recipients and formulation. My name is James. I'm the co-director of the Center with Kinam from the University of Maryland. I don't see him yet this morning. Sam Rainey, our program officer of the FDA. So we're happy of us. Welcome. So this is a 2-day workshop. But before we sort of dive in, just like to give you a little bit of background about CRCG, since I know some of you are familiar with it, others are not. This is the first time we're having a workshop on this particular topic. So this center started a couple of years ago by the FDA, in part to have some outreach to the industry. And our overall mission is to increase access to safe and effective generic drugs, to enhance infrastructure communication, which is touched on most of our education and research collaboration across industry, academia, and the FDA. So the three sort of buckets that we think of when we think about this relatively informed center involve infrastructure and communication, education and training, and collaborative research. So with regard to infrastructure and communication, this is how we spend most of our time. And I know we've spoken with many of you in the room, particularly those of you in the industry, trying to understand the challenges that you face with the particular project areas that you work in. And so here's just some of the companies we probably do outreach to at least one company every week. So for example, we did have a survey a couple of years ago, very generalized survey to understand what are the types of products and types of programming, educational programming that are most helpful. And we published that in pharmaceutical research. And then when I was on the other slide, we actually have another survey going. But what we mostly get out of these interactions are, we do take notes and we do pass some of the themes on to our FDA colleagues. And I'll just read you some comments from notes that we've collected over the last year or so that are
along with inks and structural components of adhesives for transdermals. These are just some particularly challenging Q1, Q2 formulation barriers. With respect to batch requirements, it can be very cumbersome and sometimes impossible to acquire mandated 3 batches during development life cycle, especially when these are only manufactured once a year. So perhaps this sounds familiar to you in working with complex formulations and excipients. Another topic that actually comes up quite a bit, in part related to maybe how old a product is. So variable lots of our LLDs pose significant challenge in demonstrating bioequivalence. Some of the examples that come up are iron sucrose, PLGA long-acting release formulations, which I think will be a topic today. And over the next 2 days, topical steroids were noted as not passing in vitro comparability in the hands of some of these laboratories when compared to themselves in drug release rate, particle size, purity analysis due to lot-to-lot variation. Reproducing results in meeting BE requirements with older products that have inherently variability, and the advent of newer technologies and equipment, which is of course great, but it does sometimes result in making it more challenging to actually meet initial specifications. So those are some things. So those are things that we always have on the go. Another thing that we have on the go, I think we maybe had 5 or so workshops this year. This is the final one for this year. We look forward to seeing you either in person or online. And for those of you in the room, I'll just comment, there's probably about, I think, 1,400 people that have signed up for this workshop. For those of you online, here in Maryland, we have about 120 people. So here's just a list of some of the workshops over the next year. I'll just comment for those of you who are in the audience, we do have little flyers, a little bit of a reminder, and all this is available at our website, complexgenerics.org. So in March, in a couple of months, we have a second workshop, follow-up from a workshop earlier this year on drug device combination products and the issue of substitutability. Very exciting workshop in May with regard to this newer concept of model master file. As I think you've probably heard previously, for those of you who know, a lot of you are focused on generic drug development, where maybe modeling can be an aid to development. This concept of model master file that can really help promote that. Next fall, we have one on immunogenicity for peptides and oligonucleotides. I think that's probably one of the biggest topics that I'm aware of right now. So, and later in next fall, we also have a follow-up workshop on nitrosamines. And then finally, rounding out next year, navigating the transition to low global warming potential propellants in the inhalation area. We do have an announcement. So a couple months ago, we did put out a call for a request for proposals in the research area. The call concerned adhesion performance of transdermal and topical drug delivery systems. A current approach right now is to use stainless steel as a substrate to assess equivalency of adhesion of transdermal and topical drug delivery systems. And this was a competitive award system, and the awardee is at the University of Queensland. The title of their proposal that was selected is Toolkit to Assess Adhesion Performance of Topical and Transdermal Drug Delivery Systems in Vitro. Again, we do have an industrial survey that's ongoing through mid-December, December 13th. Please feel free to visit that if you haven't already taken the survey. It's a little bit broader than a previous survey. In addition to looking at technical issues when it comes to developing complex generics, also looking at regulatory issues as well as some business type. We also have questions about business issues that can perhaps affect generic drug development. Okay, so again, unfortunately, Anna and Visha can't be here today, but Jennifer, I think you've already met her. She's at the back desk, and then Dana is online. Okay, and I think that's really about the only other announcement that I have for those of you in the audience. We do have web. Web is certainly available, and it should be free and easy to sign on. Okay, so much thanks for your participation. I do want to thank Wang and Ben Quinn, two people who put a lot of effort into today's programming. And just one feature of the programming, as you can tell from the particularly for those of you in attendance in person, a lot of effort I know that they put into putting these breakout sessions. 3 sessions later today in the afternoon, as well as 2 sessions. So today's going to be a very long day, and then 2 sessions tomorrow afternoon. Unfortunately, we can't do these breakout sessions online, but really look forward to interacting with you this afternoon and tomorrow afternoon. Have some really great discussions on what we know are some really challenging topics. Okay, so it's a great pleasure to introduce Darby Kozak. Darby, I think many of you know, he's been very active in the generic space for quite a while now. He's the Division Director for Division of Therapeutic Products 1 and ORS and OGD at FDA. I need to take off my glasses. I have some notes. Please visit our website for a more elaborate detailing of everyone's bio. We probably won't spend too much time on bios today. But Darby is, he's really, for those of you who know him, he's an expert in analytical methods, also is responsible with a team with regard to equivalency evaluation methods, with regard to complex substances, parenterals, ophthalmics, otics, and inhalation. And Darby will be giving a little introduction about the landscape. Thanks, Darby.
Yeah, thank you, Dr. Polli. As Dr. Polli mentioned, I think my background's in physical chemistry, polymers, and nanotechnology. So today's event and especially topic is really near and dear to my heart, and as well as to the advanced complex generics. I think this is going to be a really great workshop. So it's actually a real honor for me to be able to welcome all you here today to this workshop on the characterization of complex excipients and formulations. As Dr. Polli mentioned, these CRGC workshops provide us with a very unique and highly valuable interaction between both the FDA, generic industry, and leading experts in the field. So in addition to being able to share the latest in scientific research, these workshops, and really what's near and dear to me, is it enables the FDA to get a better understanding of the current analytical and regulatory challenges in the development of these complex generics, really having this interface between industry and academia to better understand what you see as the complex issues and areas where we can do improvement. So this helps us to inform not only what research is important for us to conduct, but also what may be feasible or important when we receive a drug application. So over the next 2 days, we'll hear from several experts on the importance and impact of characterizing complex excipients. From a product development and regulatory assessment perspective, I cannot emphasize more the fundamental importance of well-characterized and rational formulation. So a good understanding of the ingredients used is a critical foundation for determine a product's attributes. Developing a generic product has the same chemical composition as a reference product, reduces potential modes of failure in the bioequivalence. This is an underlying principle of why we conceive for bioequivalence may be considered self-evident for more simple solutions that are Q1, Q2 formulated as a reference product. And it's also a key reason that you often see that OGD recommends including a formulation consideration as no difference or Q1, Q2 as part of a recommendation for a bioequivalence approach. So as we'll hear more today's session, complex biodegradable PLGA materials can impact tailored extended release drug products that have been used over 30 years and in over 20 approved drug products. Although there's a long history of using PLGA in drug products, it was only earlier in this year that FDA approved the first generic product containing PLGA. This approval of the first generic naltrexone PLGA microsphere drug product is a major step in the GDUFA research program and also to illustrating that it's possible to prove generics for these complex products. As a reminder, OGD initiated its first GDUFA-funded research activities in PLGA-based products in 2013, which coincided with the very first GDUFA implementation. To date, OGD has awarded several contracts and grants, some of which you'll hear about today, to conduct research in the PLGA field. And in addition to informing FDA's current thinking and the PSG recommendations, it's also resulted in over 20 scientific articles and a number of research presentations and posters. A central aim of this research has been to facilitate the development and ultimately the approval of generics for these complex products, as generics can help reduce the medication costs for patients and create a more diverse and therefore robust drug supply chain. As we'll hear about more in today's workshop, an important part of the development and approval of PLGA products is in the characterization and understanding of what the critical properties of these polymers are. Understanding how polymer structure, molecular weight, in-group chemistry, and even the blockiness of the copolymer has been the focus of considerable research, as we'll hear about. And although gaps still remain, and I think we're going to be talking about a lot of those gaps in both the presentations as well as our breakout sessions, we've made considerable inroads into the methods to characterize these complex excipients and how those properties are likely to impact the final product's properties. With this research and success of that first generic, I'm really looking forward to the potential wave of new generics in this space. Likewise, I'm looking forward to learning more about the advances in the characterization of complex polymers used in long-acting contraceptive implants. This is another product area where we have several approved brand name drug products, but few to no approved generic alternatives. The development approval of lower cost generic alternatives for these contraceptives is a key aim of FDA, as these products can have a significant impact on women's health as well as on global health. And as Dr. Polli, and as I'm going to mention again, these CRG workshops give us the unique opportunity to candidly discuss the complexity and the challenges faced in the development of these complex excipients and products. For this reason, I'm really looking forward to today's small group discussions we're having and between our persons and attendees later this afternoon. It's extremely valuable to get this diverse feedback from industry, academia, and our regulatory stakeholders on what are the challenges being faced in the development and characterization of these products. Through these interactions, we can better understand the state of play as where we want to be, and hopefully even come up with some new ideas on how we can get there. It's through these discussions and gained understanding that we can make progress in ensuring that we're clear pathways to develop and approve generics for these products. For these reasons, I really highly commend the CRGC, the planners of today's workshop, Yan, Wang, and Ben Chen, and all of you today who are participating in this event, both today in the speaking role and in attendance. I know that this event will be packed with great information and it's going to generate a lot of new ideas to continue help solving these complex issues that are facing generic competition in the space. So with that, I'd like to thank you all in this space and welcome Yan Wang to the stage to be able to present the first session. Thanks.
Just one additional announcement. I think I maybe didn't mention it before is all this is being recorded. Slides and recordings will be available in 3 weeks. I think hopefully before the winter break. And we will reach out to you. So if you're listening to this either online or here, we'll send you an email about them being available. Thank you.
Thank you, Dr. Polli and Dr. Kozak for the nice introduction. Good morning, good afternoon, and good evening, everybody. My name is Yan Wang. I'm from the Office of Research and Standards, Office of Generic Drugs, FDA. I'm the moderator for the first session of this exciting workshop. In this session, you will hear 5 talks focusing on characterization of complex excipients that are commonly used in injectable, insertable, and ophthalmic products. The 5 speakers are Dr. Nathaniel Lynd, Associate Professor from the University of Texas at Austin, Dr. Kinam Park, Professor from Purdue University, and President of Akina Inc., Dr. Jhon, Senior Reviewer from the Office of Pharmaceutical Quality, FDA, Dr. Jafri Haley, Senior Manager and Product Specialist from Panelists, and Matthew Kihara, Senior Application Technologies Engineer, NUSO, Avental Science. Before I pass the podium to our first speaker, I would like to remind the virtual audience, please submit your questions, comments in the chat box, and we will address them as many questions as we can in the panel discussion at the end of all talks. Without further ado, let's welcome today's first speaker, Dr. Lynd.
So I'm going to give you a story kind of in 2 parts. Ultimately, we're going to get to how to characterize the blockiness, the sequence repeat unit blockiness of PLGA. First, we're going to talk a little bit about how we came up with that method. And before I really get into this, I'd like to first acknowledge my research group shown there on a fine December day in Austin. In particular, my colleagues, Mark Costello, Feng Zhang, who will present later, and the graduate student that worked on this project was Louise Kuster there in the lower right-hand corner. I'd also like to thank the FDA for their collaboration and support on this research. So polylactide co-glycolide, the constituent repeat unit structure shown down there at the bottom, and it's used in a variety of drug products. Our interest is primarily in the materials used in long-acting drug implants, so shown over there on the right. Is there a pointer of some kind? No, no pointer. Okay, I'll just continue to narrate. And so if you consider those injectable formulations in terms of the number of products that are available, there's 25 FDA approved PLGA injectable formulations here. There's, as Darby mentioned, 1 approved generic so far. And so there are a number of macromolecular characteristics of PLGA, and I'm not going to be able to address all of them, but in particular, we're going to look at the how one might establish sameness in terms of the lactide and glycolide sequencing along the polymer backbone, which does impact polymer properties. So here's a pretty complicated chemical scheme that is that underlies the chemistry that results in the formation of polylactide co-glycolide. In the upper left-hand corner there, we have forward propagation, where the different reactivities of lactide and glycolide dictate incorporation statistics. But this is an equilibrium polymerization. And so monomers add to the polymer chain, but they can also essentially fall off and go back into the mix. We also have 2 types of transesterification, both interchain and intrachain, where neighboring chains or a single chain can exchange segments with its nearest neighbor or itself. This would have a randomizing effect on the repeat unit blockiness. This is an inherently non-deterministic system. It's an unconventional chemical system. And we can understand why first by just looking at the forward propagation and depropagation and how this affects how we might model this to gain an understanding. And it turns out that because of that reversibility, you actually have to calculate the concentration of every possible sequence, which for, let's say, a polymer with a degree of polymerization of 100 would be, and you have two choices, you'd need on order of 2 to the power of 100 equations in order to model this system deterministically. That's like 10 to the power of 30 in base 10. It's a number that is so large, it just is meaningless. It's essentially infinite. So you can't write down a deterministic model for this. Now, there are good reasons why we need a deterministic model, though, so we can fit a model to data, extract parameters, and then derive molecular meaning and say, this is what the sequence of this polymer looks like. So the first step in this journey for us started with collecting experimental data. And so Luis carried out a polymerization neat using tin octoate catalyst with 16 replicants. We purged this with nitrogen, heated it up to the reaction temperature, added the catalyst, and let the polymerization run for a fixed period of time. We would then plunge these in liquid nitrogen to quench them, and then we would go characterize the composition. And so I'm showing here proton NMR spectroscopy of 3 repeats, and you can see time increases going up. You see the lactide and glycolide signals disappearing, and the corresponding lactyl and glycolyl ester sequence groups forming along the polymer backbone. And so we did these in triplicate, so we have some idea of how our experiments are producing error. Now we're looking at conversion of the copolymerization versus time separately for lactide and glycolide. Lactide is green, glycolide is blue. You'll see that glycolide very early on, it's incorporated in the polymer backbone and is essentially irreversible. So it goes to 100% conversion, whereas lactide adds more slowly, and it never quite gets to 100% conversion. And so what we can surmise from that is that that little cartoon we have there on the bottom represents the sequencing that we have in this polymer, where we have glycolide clustered near the chain initiator and lactide on the chain end. Okay, it's probably too early in the morning for this. But if we look at all of the potential reaction rates that may be occurring in this complex reaction network, we have initiation, polymerization, we have depolymerization and transesterification. And the experiment that we just did said, okay, you can ignore every equation here that has glycolide on the chain end and has glycolide reversing. So, okay, so we can cross some of these things out. And this means that now our copolymerization is governed by 3 independent parameters. Rg is the so-called reactivity ratio for glycolide. Rl is the reactivity ratio for lactide. And then we have a reversibility parameter, roughly that correlates to the equilibrium concentration of lactide monomer that will never incorporate the polymer chain. Okay, so now we have a model that we can implement. Okay, so now we can get an idea of this polymerization by fitting this model to data. We're doing regression with a numerical model. Everyone in here has probably added a trend line in Excel. There you're doing linear regression, possibly nonlinear regression. Here we're just doing regression with a computational model. We have some sum of squared residuals. That is an implicit function of all of the governing constants of the copolymerization. Okay, so first we fit the consumption of glycolide to a non-terminal model of kinetics that gives us KGG. We then fit our simplified deterministic model to the copolymerization here. This gives us Rg, Rl, and our reversibility parameter. And then we use a Brent's method interpolation to get the last constant KLL. And so based on this, now we have an estimate of every rate constant that governs the kinetics of the copolymerization of lactide and glycolide. Okay, now we had to make a lot of assumptions to do this, right? One was lactide only on the chain end. Glycolide is irreversible, and we didn't include the effects of transesterification because that is absolutely inherently non-deterministic. You can't treat it deterministically. And what we see is that there's some variation in the parameters that we determine as a function of the starting composition of glycolide. And what that tells us is that we're missing, our simplified deterministic model is missing something. And so now, but we do have a starting estimate of all the parameters. So now we move to a computational method that actually matches the complexity of the system. And that is the stochastic model regression. This is where rather than calculating anything deterministically, we do this in a probabilistic formulation where we talk about each reaction at a unit time has a certain probability of occurring. And we calculate based on that page of equations that I showed you earlier. And in this case, we're essentially doing a simulation of the polymerization. So here's kind of a cartoon of this. We have a number line that is delineated by the number of options that can happen. We can add, say, a glycolide to a lactide chain end, et cetera. We generate a random number. This selects which reaction occurs. In this case, it's adding a lactide to a glycolide chain end. We find a polymer in an array type data structure, what we can apply this to. We then increment the degree of polymerization with the lactide monomer. And then we update the time that was required for that event to definitely occur. So that is the Gillespie algorithm for simulating chemical kinetics probabilistically. And then we use that to fit numerically to our data. And so here's a sample fit of our stochastic model to model experimental data. And you'll notice that our fitting curve has noise, which is interesting, right? But that is as a result of the stochasticity of the calculation that we're doing. If we simulate a smaller solution size, then we have more fluctuations, but it is fast. If we simulate a larger, we have less noise, but it is slower. And so you have to navigate these two things as you're fitting it to your experimental data. So now we include all of the complexity of the lactide glycolide copolymerization. Our sum of squared residuals is the time-dependent conversion of lactide and glycolide. And there are 9. So we're doing multi-parameter optimization here. We have 9 parameters. So this does depend very much on where you start, but luckily our simplified deterministic model, we had a very close estimate of what the answers actually were. And how does this work? Well, it works quite well, right? We can fit routinely to reversible copolymerizations with transesterifications. It gives us a nice description of the system. We vary the starting composition of glycolide, still capture everything here. And actually, this presents a picture of what's happening in this copolymerization. So now imagine a copolymerization of lactide and glycolide. We're looking at conversion on the y-axis versus the mole fraction of unreacted glycolide in the monomer feed in the reactor. And so first imagine you're starting there at 0.25 and 0, right down there on the x-axis. The copolymerization proceeds up this conversion curve, and you see that the composition of glycolide is changing, and that's because initially we're forming a gradient early on in the copolymerization. But eventually you'll see that there's this little hook, right, where now the composition of glycolide starts increasing. And what's happening here is that we initially have that gradient that forms, but then through transesterification, what happens is eventually we enrich glycolide towards the chain end where the catalyst is, and now glycolide can equilibrate with its equilibrium solution concentration. And so what you see there is this delayed equilibration of glycolide, and the model captures that, which is really cool. So now we have our fully parameterized model for PLGA, which really gives us a view under the hood of what's happening. We have essentially an oracle now for PLGA. We can ask almost any question that we want and get an answer. And so now this brings us to the blockiness. How this is done currently is one might look at the carbon-13 NMR spectrum of the carbonyl carbon, and you're looking at heterodyads with the argument here that the more heterodyads you have, the less blocky the material is. And so we looked at that signal there at the top, and so you can see how it varies as a function of time over 120 hours. And you'll see, so first there's actually several peaks that are under there. We're not sure what they are, but it varies over the course of about 24 hours and not much after that. If you look at the glycolide-centered methylene, though, there in green at the bottom, you'll see that there's a lot, it's spread out, there's a lot more information there, and it continues to evolve over 120 hours. Another benefit of this signal is that it does not depend very strongly on stereochemistry. It's largely compositionally only. And so we set out a spread of deconvolutional fit functions so that we could separately interrogate the change in population of each of these signals and figure out what they correspond to, okay? Some experimental notes, right? You don't necessarily need to do an inverse gated experiment, but you need a long acquisition time and a high density of data points within that region is going to help you do this fit. So if we just look at these peaks as they change, we don't know what they are yet. You'll see that they change as a function of reaction time. Okay, there are some peaks that start high and go low, some that kind of stay in the middle, and there are some peaks that start at the bottom and increase. Based on the number of peaks, we thought that these are likely compositional tetrads, so sequences of 4. And so we went to our model and said, okay, tell me, how do the sequences of 4 along the polymer backbone change as a function of time? And you'll see immediately these are reminiscent of one another, not one-to-one, And so then we piece this together, mix and match to make a firm assignment here. And so these are the tetrad assignments to each contribution to the glycolide-centered methylene. The experimental data there is in the upper left, and the stochastic model prediction there is in the lower left. And so you can see that there's essentially a one-to-one comparison now. So the next test to see if this is right is to change the composition of the polymerization. This was 75-25 lactide glycolide. Now we'll change that to 40-60, and our assignments still hold up. They still survive that change in composition. Okay, so now we have these assignments. How do we create the blockiness? So now we know the population of tetrads where you have a glycolide block that is 1 unit in length, 2 units in length, 3, and then 4. And it turns out that the relationship between those is exponential there. And so we declared this blockiness function here. We have 1 minus the number of adjacent glycolides divided by sigma b. We call that the blockiness parameter. And you can see that the blockiness parameter there decays exponentially as a function of reaction time. And so what you see is that initially the polymer is most blocky because of the establishment of that gradient early, and then transesterifications randomize that copolymer sequence as a function of time. And that is how we do it. So we developed a new method of data analysis by stochastic model regression that allowed us to train a complex copolymerization model that lets us look under the hood of PLGA and to establish a chemistry-informed description of the glycolide lactide repeat unit blockiness. And how you do this is collect high-quality carbon-13 NMR spectroscopy data, focusing on the region of the glycolide-centered methylene at around 61 ppm, fit data to an array of 9 deconvolutional fit functions at fixed ppm, and then normalize the integral, sum them by block size, and fit to that function and report sigma b. And you can always contact me if you want some help with that, or you just want to hear that again. Thank you.
First of all, I'd like to thank FDA for organizing this important workshop. And let me start my talk about PLGA analytical tools for analyzing PLGA's the complex long-acting injectable formulations. Before FDA set out this program of understanding PLGA, many of the speakers already in the audience, we have studied PLGA for decades, but we never really understood what PLGA is and how we can characterize. It's only after FDA has a program we systematically understand how to characterize one by one. So whenever we characterize PLGA, we usually talk about molecular weight, and then molecular composition, LG ratio, just we heard about, an end group, whether ester or acid end group. And then we had to understand the molecular structure because one of the commercial product contains a nonlinear, the star-shaped PLGA. The question becomes when a formulation contains more than one type of PLGAs, it may have different molecular weights, it may have different LG ratios, different end group, and have different molecular structures. Then the question becomes, how do we characterize this complex formulation? And that's the question FDA had a long time ago, and we have solving the problems one by one. First, let me start with one example. Again, there are about 25 long-acting PLG formulations approved by the FDA. But one of the questions we worked with the FDA was understanding the molecular structure of sandostatin PLGA. The sandostatin has a PLGA, not linear, but star-shaped. The initiator is glucose, so there are chains, potentially 5 PLGA chains surrounding it. And there was no particular way to characterize how to understand the number of branches in the glucose PLGA molecules. So the difficulty was that, most of the time, if you look at the right side, the different initiator having different hydroxyl groups, then you can actually see how many branches are there based on NMR. But somehow glucose PLGA, the glucose initiator does not show any peaks in NMR analysis at all. And that was the problem in the beginning because we thought that there's no PLJ chains attached to glucose at all, but that was not the case. By using carbon-30 NMR, etc., the glucose, carbon-30 glucose, there are a glucose PLJ. So we had to figure out exactly how to characterize glucose PLJ. The way we used was using GPC. First of all, you separate the molecules based on molecular weight, and then the number of branching, the B, can be calculated from comparing radius of gyration, square of the radius of gyration of branched glucose PLJ compared to linear PLJ without branching. Then radius gyration of glucose PLJ, the star shape is always smaller because the multiple chains are attached to the core. So it's the radius gyration is always smaller than linear chain, and that's how we can calculate based on model the number of branching from the core. But the difficulty was that the PLJ, glucose PLJ molecular weight was way smaller than actually ideal size to measure using multi-angle light scattering. So fortunately, the intrinsic viscosity of a glucose PLJ was easily measurable and measurable in a very accurate way. So we measure the intrinsic viscosity of glucose PLJ and compare with the intrinsic viscosity of linear PLJ. And that ratio, G prime, can be related to radius gyration, comparison of the radius gyration through the parameter called drainage factors. There we can calculate exactly how many branchings each PLJ has, each glucose PLJ has. This is an example. So the way we did was using a model, glucose, not glucose PLJ, model branched molecules based on branching number 2, 3, 4, 5, 6. Now, if you compare intrinsic viscosity of sandostatin sample, you can see that branching number at the low molecular weight, branching number is about 2, 2.5, and as molecular weight increase, branching number increase to 3, up to 4, sometimes more than 4. So by same sandostatin, if you use a different lot or same lot from different time of the day or different days, they have a slightly different profile. But the main point is that as the molecular weight is lower, you have a branching number about 2 and 3. As the molecular weight increase, branching number increase up to 4. And sometimes, in a very rare case, up to 5, but usually about 3 and 4. So this is how we characterize the molecular weight. If you convert intrinsic viscosity into branching number, you can see that left side branching unit per molecule, and the bottom is the x-axis is the molecular weight. As you can see, the molecular weight is lower than branching number is around 2, between 2 and 3. As the molecular weight increase, we can see that branching number goes around 3 and 4, sometimes go up to 5. The higher number than 5 in this case is because of such a small number of molecules, the instrument cannot really measure accurately. So as you can see, most of the 90% most accurate count is around this region, where we can see that branching number is around between 2.5 and up to 4. So this is how we characterize the number of branching unit per glucose PLJ. So by using this method, we can measure a variety of different commercial products like Cobian, Lacto, Libonic. They are all within the same range as the sandostatin glucose PLJ. Another topic we studied with the FDA was TrailStar. TrailStar has more than one type of PLJ. And the question was, how can you separate each PLJs from the formulation? Now, if you look at the packaging insert, TrailStar 1 month formulation, LG ratio is a 50-50, and molecular weight is lower. But TrailStar 3 months and 6 months has a LG ratio seemingly same, 75 or 25 range. Molecular weight is about the same too. But the question is, the 3 months formulation and 6 months formulation cannot have the same PLGA, because if you simply double the amount of drug, it doesn't mean that you can release twice longer. Okay, the initial burst release may be twice higher, release may be twice higher, but duration may remain the same. So the question is, how can you characterize different types of PLGA that may be present in a trail cell formulation? There we found that, PLGA range LG ratio from 50-50 all the way to 110. They have the same name, PLGA, but they are not exactly the same polymers. Now, the left table, the right side figure, they are exactly the same information, but it's easier to explain using table here. For example, let's talk about benzyl alcohol. Benzyl alcohol, the white area means it doesn't dissolve. Benzyl alcohol does not dissolve PLGA 50-50 all the way up to 70-30. But PLGA LG ratio 75-25, benzyl alcohol dissolves. If two polymers do not dissolve in the same solvent, what does that mean? Two polymers are different. So PLGA 70-30, 75-25, seemingly same PLGA, but molecular nature, they are very different. They have a different properties. The same thing goes to other ratio too, like ethyl benzoate, 60-40 dissolve, does not dissolve, but 65-35 dissolve. So what this shows is that PLGA, even though LG ratio is seemingly a little bit different, but they become totally different polymer, partly due to, because of the blockiness we talked about in the first talk, partly due to the blockiness differences and many other reasons, they are different polymers. So first thing we need to understand is PLGA, they may have the same name, but they are very different polymers. Okay, that's the first thing to understand. Now, over the years, working through FDA, we spent enough time to screen larger number of organic solvent and their mixtures. So now we can separate PLGA based on LG ratio. For example, 61 loctite contents and 63 loctite content, we can separate them. Okay, that's the beauty of this study supported by FDA. We can separate PLGA regardless of the LG ratio. As long as they are different, we can separate them. So having all this, separation and characteristics that we went through, we noticed that there are at least three different groups. First group, LG ratio is about 83-17, and then the other is about 73, the other is 70. Now the question becomes here, how do you distinguish the LG ratio 73-71? The reason is that we measure the blockiness using NMR, then you can see the group, the first three has a distinct blockiness about 0.4, which is very different from the next one, about 8.5. And that is also different from blockiness 1. So we can confidently say there are three different types of PLGA present in the trail cell formulation. So in the end, we conclude that there are three different LG ratio. One is 85-15, 75-25, 70-30. I mean, this is the beginning of the formulation, because we can say that I don't think that any, if standard setting manufacturer purchase, they will probably purchase 85-15, because as far as I know, no manufacturer make 83 or 27, 17, etc. So the bottom line is that these are raw materials, but then what we need to do is characterize the pH composition of the final formulation. This is why FDA set out to develop a number of different assay analytical tools to isolate the PLGAs and understand what PLGA they are based on the final formulation. Okay. Now, not only component, the PLGA type, amount, end group, LG ratio, type of solvent used, drug, etc. Those are affecting the final formulation properties, but also important is the processing. Even if we use the exact same components, depending on how we process, the final formulation will be different. The final drug release kinetics will be different. And this is what is showing. They're a little bit complex, but the point is that not only composition, but manufacturing process will affect the final properties of formulation. As you can see here, depending on how we make it, some microparticle can be smooth, surface can be smooth, sponge, or porous or wrinkled. All these are the result of processing. So by studying the exact composition and the appearances, the surface morphology, we can kind of guess what kind of processing was used. Also, again, with the support of the FDA, we studied how to go a little bit deeper than the surface. And there we analyzed the inside of the microparticle by using so-called semi-solvent vapor method. We can actually selectively remove the surface PLGA and then see it inside the core. So this is one example of I think Vivitrol. If you remove the surface PLGA layer, you can see that nitroxane crystals accumulate in the center. And that's why after initial burst release, there's a little lag time for several days. And probably this is one of the reasons. This is the Vivitrol. This is another formulation we made in the lab. So finally, the current project with the FDA is, this is all fine. We can separate PLJ and characterize each one of them. The question is then, even if we have not enough PLJ, can we still characterize them? For example, Durysta has a total weight of less than 100 microgram, including drug. Total amount of PLJ is less than 100 microgram. The question is, can we characterize? Of course we can. If we buy hundreds of them, thousands of them, we can have enough PLJ. But the question is, who's going to pay all this? Because each formulation costs thousands of dollars. If you buy 100, the cost is prohibitive. So the question is there any way to characterize? Even if amount of PLJ is not enough in a given formulation, and even if a small amount of PLJ formulation have more than one type of PLJ, can you still analyze? That's the key question. So one thing we need to do is to increase the sensitivity of the PLJ itself. To do that, we can add some kind of a UV fluorescent probe, or as Darby mentioned, our chairman here, he mentioned that not only fluorescent probe, but some UV chromophore may have enough signal, especially in a different semi-solvent conditions. So we are exploring all those. And the question is that how do you separate different polymers? For example, if formulation has a linear PLJ and star-shaped glucose PLJ and different LG ratios, different molecular weight, all at the same time, a total amount is less than 100 microgram, how can you separate them? And that's where we came up with the temperature-controlled sequential semi-solvent gel formation chromatography. By using several different semi-solvent, we can separate, first of all, PLJ based on LG ratio. And then by using different selectively, carefully chosen semi-solvent, we can see that different LG ratio having different, hydrodynamic radius gyration or intrinsic viscous changes. So we can use that to find out exactly what LG ratio is. And also by using radius gyration measurement, we can see that the information about linear or star-shaped polymers. So this is how we are preparing right now to understand how to understand the exact composition of a formulation like a Durysta. Again, total amount is less than 100 microgram. Now, Ozurdex, I think another speaker to Feng Jiang will talk about this in detail, but this is, again, composition is known, but as far as we know, nobody has analyzed the final actual formulation. This is information from the manufacturer. The signal for also has another interesting information, because it has not only glucose linear PLJ, but glucose PLJ starch A polymers. And the LG ratio is about the same. And that's where we need to figure out exactly how to separate them and characterize. So all these things, effort is to characterize this small, sample, which we simply call a mini-sized complex formulation. Then other generic companies or other scientists can have reverse engineering to understand exactly what PLJs are there. But as a scientist, the more important question to me is, why any company is making such a complicated formulation? Is that the only way to achieve the release kinetics they want to? If a simpler formulation cannot do that, that's the kind of ultimate question that I'd like to solve too. So if the simpler formulation can have a drug release kinetics exactly the same as complicated formulation, the question is, why do we have to make a complex formulation? I think that's the ultimate answer we should provide, and that will also help the FDA to approve the generic formulation, which may not be exactly the same as a complex formulation, but essentially does the same thing, same release kinetics, same efficacy, etc. So that's my talk, and obviously there may be some questions. We'll talk about this in a later time. Thank you very much.
Good morning, everyone in here, and good morning and good afternoon, good evening in online participants. Okay, first, this is an OPQ standard slide. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. Okay, from the last two talks, you just using the lots of brain for mathematics and chemist. And this time, just a little bit relaxed, and then what is the regulatory perspective for the PLGA product. So we just focusing on the generic PLGA drug product ANDAs, common deficiencies and expectations. Okay, this slide is my disclaimer. And today's my purpose for talk is I want to share common issues observed during regulatory assessment of generic drug product which contains complex polymeric excipient, which is a PLGA. And also, I want to help you to develop generic PLGA drug product. Okay, in this slide and next slide show the FDA-approved PLGA-based product available in current market. There is two types of drug substance using for this PLGA drug product. One is small molecule, and the other one is peptides. Okay, first we can see that in the small molecule, as you can see, most of drug products are intended for parental use, except Arestin in this list. And also you can see that in the right column, all of the labeling described in the brief PLGA characteristic, except Arestin and Ozurdex, which is in the lactide to glycyrrhide ratio. So just remember, lactide to glycyrrhide is one of the key components to evaluate for PLGA sameness. I'll discuss it later. And also, one of the interesting part is Ozurdex is very clearly described in their labeling for the PLGA commercial name. This is very unique, which is Resomor, RG502, and 502H. Okay, compared to the previous small molecule-based drug product, peptide-based drug product also, most of them are intended for parental usage. But if you see in the right column of this table, half of them doesn't have PLGA characteristic information. That is also in the back challenge at the beginning. But fortunately, Eliad giving the labeling, giving another information, which is a polymer end group information. And also in the previously Dr. Parks in the talk, the sandostatin LALD part giving the polymer PLGA structure information, which is a glucose star polymer. Okay, from the previous two slides, we just know about the most of the drug product are intended for parental usage. And so per 21 CFR, generally a drug product intended for parental use must contain the same inactive ingredient, which is qualitatively or Q1, and in the same concentration, which is quantitatively or Q2, as the reference listed drug identified by the applicant. So now is how we can demonstrate qualitative sameness for PLGA. At the start with most of the labeling described of the polymer composition, as Dr. Parks mentioned about, we just said PLGA, but PLGA itself is different. And so we have to figure out lactide to glyceride ratio by carbon-13 NMR and proton NMR. In the bottom of in the left side and middle side has typical spectra from the proton NMR and carbon-13 NMR, but I'm not detailed to dive in because already mentioned the previous two talk using for the how to evaluate lactide to glyceride ratio, and also in the first talk of the Dr. Lynd said about how to figure out blockiness. And also you can see the earlier labeling. There is a polymer end group information is important. And so you have to demonstrate what kind of the polymer end cap of PLGA. And also PLGA is a sort of the polymeric material. And so as the same evaluation of the general polymeric science evaluation, we need weight average molecular weight and number average molecular weight with polydispersity index, and also glass transition temperature and viscosities. There is two kinds of the viscosities. One is inherent viscosity. The other is intrinsic viscosity. Intrinsic viscosity is determined by measuring the relative viscosity at several different concentrations and then extrapolate the specific viscosity to zero concentration. From previous Dr. Park's talk, and also in the sandostatin LALD pass labeling, there is some of the PLGA we using, the drug product using for the structural PLGA. And so now we question for how we can demonstrate qualitative sameness for PLGA structure. Before starting talk, the left top corner of this slide, actually there is Dr. Park's slide talk also using this cartoon, but I'm just a little bit briefly touch for this concept. There is in the three cartoons, left and right one has a linear PLGA, and the middle red cartoon is a star shape of the PLGA. The big difference is star PLGA has multiple linear PLGA chain anchored to the core. So due to this feature, and also is another static hindrance for the moving of the linear chain, there is something is different. One is compared to first and second one, there is a similar molecular weight, but hydrodynamics volume of the linear PLGA is much larger. And oppositely, you can see in the middle one and right one, that looks like in the hydrodynamic volume is pretty similar, but molecular weight of star PLGA is much larger than linear PLGA. And so it's a conventional size exclusive chromatograph can solely characterize the PLGA characterization. And so we need some more information also in the lots of idea from the Dr. Park's talk. The first, hydrodynamics radius calculated by the hydrodynamics volume, and also radius of gyration. What is the radius of gyration is previously Dr. Kinam said, root mean square distance from the center mass. And so if it has more polymer chain close to dense in core, there is in the radius of gyration is smaller and smaller. And also there is another need to do more information for how many arms attached to the core and how they are standard deviations. So here is an example for the marked efficiency for PLGA qualitative sameness. PLGA properties may be sensitive to microsphere manufacturing process, and your submission is lacking in experimental characterization data for the PLGA polymers in your test product and RLD product. To demonstrate your proposed polymer is the same as polymer used in the RLD product, we recommend you provide comparative characterization data for the PLGA polymer, extract at least 3 batches of the same strength of the proposed test product and RLD, including the following tests: polymer composition, which is lactide to glycolide ratio, and 2 molecular weight with polydispersity index, polymer end cap information, glass transition temperature, inherent viscosity, intrinsic viscosity, radius of gyration, hydrodynamic radius. For the detail, you can get this publication at the bottom. Also, additionally, how we can qualitative sameness for PLGA structure. This is another example. Please provide the following comparative physicochemical characterization data for the glucose star PLGA copolymer extracted from at least 3 batches of the same strength of the proposed test product and the RLD. There is an additional test. The isolation method for star PLGA extraction, number average molecular weight by osmometry, presence of glucose and distribution in star PLGA polymer, detailed calculation for degree of branching, which is number of arms, standard deviation of number of arms. Also, you can get the more idea from the previous Dr. Park's talk or this publication on bottom side. Last 2 slides, we're discussing about the qualitative sameness for excipient itself. Now we move to the qualitative sameness for the finished product. Here is an example of mark deficiency. We recommend that you provide comparative data for the drug product microspheres, at least 3 batches of the proposed test product and RLD, including the following tests: molecular weight, particle size distribution, surface morphology, in vitro release, drug loading, porosity, water content, assay, impurities, and content uniformity. We expect you to provide detailed methodology for comparative physicochemical tests and same age for all experiments. Also, we recommend that you provide representative scanning electron microscope images at various magnifications to allow better assessment of the microsphere surface morphology. Okay, now we just move to the manufacturing part. Today, I'm not touched to detail for how the PLGA manufacturing process itself and how the various of the synthesis route of the PLGA. And this slide is the same as Dr. Park's in the talk. There is in the multiple parameters can affect to the PLGA physical chemicals characteristic properties and its structure, followed by drug release kinetics. But one of the important part from this manufacturing process, if you can see the lot of organic process solvents are using during the drug product manufacturing process. Okay, before starting the drug product manufacturing process, and PLGA manufacturing process is very complicated, and there is a multiple synthetic route. And currently we using the ring opening polymerization. So we recommend that all relevant information for PLGA manufacturing process and control be provided through Type 4 DMF or ANDA submission. If all information provided through the Type 4 DMF, just remember, PLGA DMF should only focus on excipient physical-chemical properties, manufacturing process, and controls. PLGA sameness demonstration should be provided through ANDA submission. Here is an example of the mark deficiency. Please provide detailed information on synthetic route and manufacturing process for your proposed PLGA excipient, including the in-process control for each manufacturing step and control of starting materials that are inputs to manufacturing process. Also, you can provide all information directly in your ANDA or by your excipient manufacturer in the form of a Type 4 DMF, which is then referenced by your application. As previous manufacturing slide, I mentioned about the loss of organic processing solvents are using during the drug product manufacturing process. Therefore, you should consider the risk of contamination of drug product with leachable from the manufacturing equipment. And so we recommend that you consider the potential for manufacturing process leachables or possible impurities from unknown sources, such as processing solvents, to be trapped in the drug product microspheres in the risk assessment used to design the extractable and leachable studies for your proposed product. Please discuss how you detect and quantify leachables or possible impurities trapped in the PLGA matrix and provide evidence to demonstrate that the sample preparation procedure for your analytical method is adequate to quantify all such analytes in the drug product. The sample preparation procedure should be able to fully solubilize your drug product sample. Also, there are lots of organic process solvent using during the manufacturing process. Another example of mark deficiency related to the residual solvents. Because residual levels of solvents used in drug product manufacturing process may significantly affect drug product quality. We recommend that you monitor the levels of all solvent used during the manufacturing process in the finished product. We recommend that you justify adequately the proposed limits for these residual solvents. The justification should follow the recommendation outlined in the ICH Q3C guideline. All right, that's all I have today. And thank you for your attention. I'm happy to take any question at panel discussion later. Thank you.
All right, so good morning. My name is Jeff Haley. If you want to get in touch with me, my email address is on this slide. So if you have any questions, you want to contact me, you want the slides, whatever, just send me a note. So today I'm going to talk about ethylene vinyl acetate copolymers that are used in long-acting drug delivery systems. Now, EVA was first commercially introduced in the 1960s and has been used in drug delivery systems for a very long period of time. Today, though, our focus is really going to be on understanding what's important and relevant about the material for looking at some of the more recent products that are still in the market today, specifically contraceptive vaginal rings and long-acting subcutaneous implants. So to kind of, there we go. So to kind of motivate this talk, right, I think we want to think specifically about this sort of a system. We want to think about a subcutaneous implant that's a multi-layer structure, and that shortly after implantation, there's a short burst release of the hormone from the implant, followed by a very sustained delivery of the drug over a long period of time. So this is the type of system we'd like to understand. So let's look at it in a little bit more detail, and then we'll jump into the polymer chemistry. So in a system like this, you typically will have a core that's highly loaded with drug, right? And that core is EVA plus drug. And then it's surrounded by some sort of rate controlling second layer that functions as a membrane. And so you actually can manipulate the release characteristics of a device like this by adjusting parameters such as the thickness of that outer membrane, as well as aspects of the chemistry characteristics of the polymer that's used in that membrane. So that brings us to what we're going to talk about today. We're going to talk today about some of the characteristics of ethylene vinyl acetate copolymers, which is shown here. The structure is a copolymer of ethylene and vinyl acetate. So we'll first talk a little bit about the polymerization, molecular weight effects, and how that might be indexed and characterized. We'll talk a bit about vinyl acetate content. That's a very, very critical parameter for describing the drug release characteristics of the material. And then we'll spend maybe the last few minutes of the talk diving into some of the more complex aspects of this polymer, which is around the branching effects that occur in the chemistry. So at a very simple level, the chemistry is pretty straightforward, right? You take ethylene gas, you take vinyl acetate monomer, you bring those two things together, you compress them to an almost alarmingly high pressure, introduce peroxide initiator, and you get a very rapid exothermic polymerization of a copolymer. But anyone who's studied free radical polymerization knows that there's a lot of different things going on. And so it's actually the relative rates of these various reactions will define kind of what the polymer structure is, what the polymer architecture is, what its molecular weight will be. So these are all very, very important. And at a practical level, you control these relative rates of these various reactions by controlling things like reactor temperatures, feed gas temperatures, reactor pressure, and a variety of other parameters that are blocked in the product manufacturing specification. So the first thing you want to characterize with the polymer is the molecular weight. And we'll talk more about using things like gel permeation chromatography in a minute. But actually, the method that's typically used to do product release and product specification for a material like this is something different. It's a melt index. So this is done for a variety of reasons, but not the least of which is it's a very fast, reproducible, robust method. So what you're doing is you're taking advantage of the fact that as a polymer molecular weight increases, its viscosity also will increase, right? So when you do a melt index measurement, you essentially take the polymer, you heat it up, and you put it in a barrel, and you compress it under a known load, and you force it through an orifice of a controlled size. And by measuring the flow rate through that orifice, you're able to extract information that you can relate to the molecular weight of the polymer. So again, the nice thing about this is it's very reproducible measurement, very robust. If every laboratory should be able to get almost the same exact measurement on this kind of a test. So the parameter that becomes very important for understanding long-acting drug delivery systems with EVA is actually the vinyl acetate content of the material, or the ratio of the ethylene and vinyl acetate in the polymer itself. And typically this is going to be reported on a mass basis of vinyl acetate. So the reason why this is important is that polyethylene is a material that readily and easily crystallizes quite rapidly to a quite high degree of crystallization. By introducing vinyl acetate into the polymer backbone, the ester side group functions as sort of a bulky defect that disrupts and frustrates the crystallization and suppresses crystallinity of the material. So as you introduce more crystallinity, more vinyl acetate into the polymer, you see the crystallinity will drop. So if you made, say, like a low-density polyethylene homopolymer, you might nominally get a crystallinity of 50%. But as you increase vinyl acetate, that drops down to eventually 0 until you make an amorphous polymer. And this is going to be very, very important for understanding release kinetics of an implant such as this device. And that's because the drug, the characteristics that matter for the drug to escape through this device, if it's going to escape radially from this multilayer implant through that outer membrane, it has to dissolve and diffuse through that membrane. So the solubility of the drug in the membrane and also the diffusivity of the drug through that membrane layer is going to be a function of how much amorphous content is in the material. Right? Because you can't diffuse, these molecules will not diffuse through the crystals. The kinetics are not sufficient for that to happen. So here's an old example that we did in our laboratory a number of years ago, where we just wanted to develop some very basic demonstration data on how both melt index and vinyl acetate content can affect drug release characteristics. So we took niacin, simple, easy to handle model compound. We put it in, did extrusion and put it in at 5% by weight into EVA. And if you look at this plot, you see there's 3 different colors of curves, right? So we're showing the amount of drug release versus time. And you can see there's the green curves, which correspond to a 9% vinyl acetate copolymer. There's the red curves, which correspond to a 28% vinyl acetate copolymers. And then there's the blue curves, which correspond to 40% vinyl acetate copolymers. The other thing that you can see is if you look closely, you'll see if you compare the 2 red curves we actually did 2 different copolymers in this. We used a 3 melt index and a 25 melt index copolymer of 28%. You see there's essentially no meaningful difference in the release characteristics of those two. And likewise, if you compare the two different molecular weight or two different melt index versions of the 9% copolymer, so again, see a very similar release characteristic. So that was a sort of a monolithic system where you just force the drug to dissolve and diffuse through the polymer matrix and you characterize its release. Here's just another really simple example that we did in the laboratory with naproxen. Again, easy to handle drug. It was a kind of a project we can give to a summer student who we don't have to train high potent handling techniques to. So in this case, if you take a 28% vinyl acetate core that has about 10% naproxen by weight in it, you see the release characteristics of that without a membrane. But then as we attach different vinyl acetate copolymer membranes to that core, we see that we get very different release characteristics from that. So with that, I mean, vinyl acetate is going to be a really, really critical parameter for the material. So we have to be pretty thoughtful about how we characterize it. So the standard way that you're going to see people characterize vinyl acetate content for product release, for product specification purposes, is going to be with some sort of FTIR method. There are standard methods that are published that no one essentially uses. So in practice, everyone has their own method that they've validated and they've used for, in our case, 40 years. So the method's very robust, but if you go from one manufacturer to another, the methods won't necessarily be the same. The other thing is these methods use solid samples. So that has some things that the sample preparation can in principle affect the results of the measurement. So again, the sample preparation has to be part of the validation, has to be carefully validated to make sure you get the right results. But on the other hand, this is a very fast method to run. It can be run quickly, many times. It's really good for product release and good for product specifications. There are other ways to characterize vinyl acetate, ranging from challenging and esoteric to, I think, more routine. Obviously, pyrolysis is a practical method to do this, or it's a method you can use. It's actually a very challenging method in practice. But in this case, you degrade the ester, right? So you heat up the polymer to a temperature where the ester decomposes, and you can characterize weight loss, or you could attempt to capture the acetic acid that's generated in titrate. So that is a method that's been used. I think a more practical one to consider here is probably proton NMR, right? So again, many laboratories will have an NMR in it. You can develop methods here based on comparing, the relative intensities of various peaks in the structure. The other nice thing is that in proton NMR, you can get a peak that's for the CH3 end groups that are associated with the polymer, which is going to give us some, we'll talk about that more in a little bit when we talk about branching of the polymer, because that allows us to characterize the branching of the material. Okay. So last piece on copolymers, there was a very nice talk earlier about in PLGA, the effects of copolymerization and how you have to deal with the reaction kinetics of PLGA polymerization. With EVA, the story is actually quite simple, right? We're always considering, when we talk about a copolymer, we think about the reactivity ratios. The reactivity ratios tell us something about the likely blockiness of the polymer that we're going to achieve. One of the nice things about EVA is that for the polymerization of ethylene and vinyl acetate via free radical polymerization, the reactivity ratios are very, very close to 1. So you have something that's relatively almost exactly a random copolymer. So from the standpoint of characterizing blockiness and things like that in the polymer, or the relationship between molecular weight and the copolymer content, those should be fairly uniform across the polymer. It's just baked into the chemistry. So, okay, so thus far we've talked about all the, we've kind of created a fairly simple picture of what this material is like and how it's characterized. So now we have to jump to the part of EVA that makes it more complex. And that comes from, if you recall, I put up a slide earlier of all the different mechanisms that you can encounter in free radical polymerization. One of those is chain transfer. One of the complexities of ethylene vinyl acetate copolymers, and in fact, all ethylene copolymers in free radical polymerization, is there substantial chain transfer to the polymer itself in the reaction medium. So this can happen in two forms. First of all, there's intra-chain transfer. And so in the illustration here, if you have a chain with a growing free radical on the end of it, there's potential for a backbiting reaction. And then you grow from there off of that point and leave behind a short branch in the polymer structure. And so short chain branching, there can be quite a lot of this. And I'll show some examples of roughly how much you get in a polymer here in a minute. The second form of branching that occurs is long chain branching, and this comes from interchain transfer. So essentially, if you have a concentration in the polymerization medium where you have a meaningful concentration of dissolved polymer in the same solution with growing radicals of polymer, you can get transfer from one polymer to another, and that introduces now a place to start polymerization at some point along the polymer backbone. And as the polymer grows off of that, you'll create a long chain branching architecture. So we have to concern ourselves with both of these things. So first, let's talk about branching in general. Here, when I'm talking about branching, the majority of the branching in the polymer is going to be short chain branching, but there is some long chain branching that'll come up in this number as well. So first of all, much like vinyl acetate, these branches are bulky side groups that don't readily incorporate into polymer crystals. So much like the effect of vinyl acetate, increasing the amount of branching in the polymer is going to suppress the crystallinity of the polymer. Now, the thing is we can tune the amount of branching in the polymer a little bit. We can adjust things like reaction pressures and move it a small amount. We can't tune it to the same extent that we can tune the amount of vinyl acetate we incorporate. If we could, we wouldn't need vinyl acetate in the material to give a material with all these different crystallization or crystallinity characteristics. But what it does tell us is that we're going to have some branching, and that's going to have some effect on the crystallinity of the material. So here's an example, again, taken from our laboratory where we took a 28% vinyl acetate copolymer. We used an NMR method where you essentially determine how many N groups are in the polymer. And if you have some knowledge of the molecular weight of the polymer, you can infer or estimate the number of branches per 1,000 carbon units. And so you see in this example here, there's about 15 branch points per 1,000 carbons, and there's about 50 acetate units per 1,000 carbons. So you see that there's a lot more acetate units than there are the branch points, but there's still a significant amount of branch points, and those are going to impact crystallinity. The difference is we can tune the acetate units from 0 to 100 per 1,000 carbons, and we can only move the branch points a small amount. So we can't use that as a practical tool for changing the release characteristics that you get from a system, but it will have an effect. And a practical consequence of this is potentially two different materials with the same vinyl acetate content could have somewhat different crystallinities. So now let's go into long chain branching, and this is where things get a little bit more hairy still. So thankfully, I follow a few presentations where people have looked at some of these comparable issues in PLGA. So there's a variety of ways that one can go and try and attempt to characterize the long chain branching and the long chain branching in combination with the polydispersity of the material. So first of all, you can use GPC measurements as were described briefly, and you can use, these are nice in the sense that they describe a molecular weight distribution in a way that we can understand, and that you can gain insight about the branching by doing some of the things that have been described already today. But for example, you can take measurements of, if you have a multi-angle light scattering detector as a part of your GPC system, you can make measurements of the radius of gyration as a function of molecular weight, and the relationship between radius of gyration and molecular weight will tell you something about the branching, the long chain branching of the polymer. So that's nice. The downside of this method, there's a couple. One is that highly branched, high molecular weight polymers can co-elute in a size exclusion chromatography experiment with other less branched, lower molecular weight copolymers, just because this separation method is primarily due to hydrodynamic volume. The other thing is that these instruments can be challenging to maintain, and lab-to-lab reproducibility is not typically very easy to achieve. So an alternate method, and one I'll talk a little bit more about here briefly, is to use oscillatory areology. So this is very robust, very easy, very fast to do. It's very reproducible. And also, it's nice because it is most sensitive to the types of effects of long chain branching that will ultimately impact the rheology, processing characteristics of the polymer, and then how that plays into how drug release will occur from a system. On the other hand, the measurement's indirect, so it's hard to visualize what this difference is. So I think we look at it more as we're developing an index of these sorts of characteristics in the material. And then you can do other more exotic things, like people have studied long chain branching with small angle neutron scattering, for example. All right, so let's talk really very simple level, and I'm going to go very, very quickly through this, but I will leave a reference in here. I'm happy to talk more in detail about this if anyone's interested. So if we have a complex mixture of polymers that are polydispersed with varying degrees of long chain branching, any mixture of polymers will produce some linear viscoelastic mechanical relaxation spectrum in some sort of linear viscoelastic measurement. And any spectrum of linear viscoelastic relaxation times will in turn produce linear viscoelastic measurement data like an oscillatory rheology measurement where you measure storage and loss modulus as a function of frequency. So if we can infer things of what we'd like to do is use that oscillatory rheology measurement to infer things about the breadth of this mechanical relaxation spectrum. And then if we can compare things about the breadth of that relaxation spectrum, that gives us a basis for comparing different polymers. There's a lot of different choices you have, but a simple choice for this is to look at the value of the storage modulus at a particular value of loss modulus, where that loss modulus is select to the point where you're very, very close to the terminal regime in the linear viscoelastic measurement. And like I said, it's a little outside our scope to talk about why this is today, but let's accept this for the moment. And there's a nice reference here from some folks I used to work with a number of years ago. So if you look at in a measurement like this, you might pick a value of say like 500 pascals for G double prime. And then you could compare the value of G prime at the same frequency across a number of polymers. And then this will give you a very sensitive measurement that can then be used to actually predict some of the other elongational and rheological phenomena that you'll see when you start to actually process the material. So let's talk now why we might care about that. Hot melt extrusion. So these implants are typically made through some sort of a hot melt extrusion process where drug is compounded with polymer via a twin screw extrusion. And then there's some subsequent co-extrusion process where you make the final implant. And so if you go to the literature on this, you'll find that there's big dependency. There can be effects of processing conditions on drug release characteristics. And so this is a nice publication from about 20 years ago where they varied a lot of process parameters, and they showed that the release rate from a multi-layer implant was function of the drawing force that they measured, which is really a measure of the orientation that you're getting in that polymer during the extrusion, drawdown, and cooling process. Well, this is where the long chain branching and melt index and rheological measurements could impact drug release, right? We said initially vinyl acetate content and crystallinity is the primary effect here, but because you do have potential to have orientational effects, if you had very, very different polymer architectures and you put them through the exact same process, you might end up in a situation where you have different drug release characteristics as well. Now, you can probably fix that on the back end by adjusting processing parameters, right? And no two processes will be exactly the same. So there will always be some work to optimize this and dial in the release characteristics based on processing parameters. But this is an aspect of the polymer that's going to play a role in that. So to wrap up my summary here, so EVA is an essentially random copolymer of ethylene and vinyl acetate. And the two key characteristics are going to be melt index and vinyl acetate. That's how products will be specified. But we also have to worry ourselves with other aspects of chain architecture and how they're going to interact with the processing of the polymer when you actually make the final drug product. So that is, I believe, all I have.
Sorry about that. My name is Matthew Kihara. I am a senior applications engineer for NuSil covering silicone materials used for implantable and drug delivery applications. For the next 20 minutes, I will be taking you into the world of silicone excipients used for implantable drug delivery applications and the common types of characterization used for their bulk material properties, their physical properties, and their chemical and biological properties. Before we begin, it's important for us to review this disclaimer. The information presented is intended as a general overview only. All purchasers and users should conduct their own testing and regulatory review to satisfy themselves as to the appropriateness of these materials for their specific needs and uses, as well as with respect to regulatory and compliance requirements. Can you please go back one slide and move when requested? Thank you. All right, the NuSil brand. Next slide. Now I'd like to take a moment for those who are not familiar with NuSil to quickly introduce the brand. Avantor's NuSil brand is a trusted global partner and silicone supplier to customers in the life science and advanced technology industries. We are a market-leading manufacturer of ultra-high purity medical-grade silicones for a wide range of medical devices, medical and cosmetic implants, and drug delivery devices and implants. NuSil maintains over 700 master access files and drug master files with the US FDA. Next slide. This presentation will start with a quick overview of the many types of silicones used in the medical and drug delivery industry. Then move to regulatory support. Then we will discuss the common types of characterization used for their bulk material properties, their physical properties, and their chemical and biological properties. And then finish with additional areas of consideration. Again, will you please go back, I believe, 2 slides now, and only move when requested? Thank you. One more, please. Forward. Yeah. And now the first of 4 sections, silicones used in the medical industry. Next slide. You all have likely noticed that silicone is used throughout the healthcare industry, both inside and outside of the body. Some common examples of this are pacemakers, breast implants, intrauterine and intravaginal devices, IV catheters and tubing, and various implantable shunts and valves. Each of the items depicted on this slide has unique material, chemical, and biological requirements, which are essential to the performance, safety, and success of the device. For this reason, it is important to evaluate the application to identify the crucial properties, chemistries, additives, and regulatory requirements needed for success. Some common examples are the flex fatigue resistance of a finger joint, or the bond strength and cure chemistry of an adhesive used to assemble a pacemaker, or the elution rate and reactive chemistries of a hormonal active pharmaceutical ingredient in an intravaginal ring. Next slide. Silicones are unique as they not only come in a wide variety of reactive chemistries, but also come in a wide variety of forms. Each product contains a unique blend of multiple reactive and unreactive silicone polymer components with different molecular weights, functional and reactive group types, concentrations, and locations. And they also include various functional and non-functional additives and fillers. And so it is very rare that any 2 grades of silicones will be chemically and physically the same, which may lead to unforeseen issues when a developmental generic device is implanted into the body. In fact, due to this wide variety, NuSil provides over 700 master file containing products for use in long-term implantable and drug delivery applications, which have undergone biological, chemical, and physical testing aligned with relevant ISO and USP standards. This slide shows a high-level overview of some of the many forms in which implantable and drug delivery grade silicones come in, including primers, adhesives, coatings, colorants, inks, foams, resins, gels, fluids, greases, elastomers, and dispersions. The elastomers, which many of you are familiar with due to their use in injection mold parts or extrude into silicone tubing for many types of medical devices, contain liquid silicone rubber, or LSR, and high consistency rubber, or HCR. All these groups come in a wide variety of reactive and unreactive chemistries and contain various types of additives and fillers used to meet device-specific performance and safety requirements. Next slide. Our second section, regulatory support. Next slide. At the most basic level, regulatory support is providing specific information about a material in a clear, concise, and contextualized manner to help customers and regulatory authorities determine that a material is safe and suitable for the customer's specific end use. For this reason, performing robust material characterization is a foundational component of NuSil's regulatory strategy. The more we know about our materials, the better positioned we are to respond to and support customer and regulatory questions. Next slide. Our third and most in-depth section, types of characterization. Next slide. As part of product and device development, it is important to establish the material's typical bulk material properties, physical properties, chemical properties, and biological properties. Information on the bulk and physical properties of a material help customers and regulatory bodies to determine if a selected material can meet the performance requirements of the specific end use. Chemical properties are used in combination with the biological properties of a material to help customers and regulatory bodies determine that a selected material does not pose unacceptable risks to the health and safety of the user and patient. Much of this information can be found within the silicone material's master access file or drug master file. Multiple ASTM test methods exist to cover these characterizations. Any areas where differences are found between a generic and the original device's materials should be flagged for further investigation. Now we will go into each of these 4 segments to review the typical characterization involved for each. Next slide. Regulatory bodies want to know what substances and compounds are used in medical applications for which they have oversight. Accordingly, they will want to know the chemical composition of the silicone products in the form they will be in as the component of the end application. If the product cures into a solid, they want to know the composition of the cured solid material. Composition includes impurities, additives, and processing aids. Areas where notable differences are found between a generic device and the established device's materials should be flagged for further investigation. Some typical key items of interest for bulk material property characterization of silicones are chemical structures and composition, chemical cure chemistry. Some common ones for silicones are platinum, tin, peroxide, and UV. Byproduct and residual composition from the curing mechanism and processing, appearance of the cured and uncured material, odor against the standard, particulate contamination of the cured and uncured material, the molecular weight and degree of polymerization of the uncross-linked polymers in uncured form, and trace metal analysis. Again, multiple ASTM methods, ISO standards, and USP standards exist to cover these characterizations, either generically or for specific end uses. Next slide, please. Information on the physical properties of a material help to allow customers and regulatory bodies to determine if a selected material can meet the performance requirements of the specific end use. Again, areas where notable differences are found between a generic device and the established device's materials should be flagged for further investigation. Some typical key items of interest for physical property characterization are durometer or hardness of the material, adhesion strength for applications where adhesion or bonding are needed, filler distribution within the polymer, the tensile strength, elongation, and tear strength of the cured material, modulus at various percent elongations, tensile set at representative elongations, compression set of the cured material, cross-link density of the cured material, volatility of the cured material, and the specific gravity of the cured material. Again, multiple ASTM test methods, ISO standards, and USP standards exist to cover these characterizations, both generically and for specific end uses. Next slide, please. Chemical characterization data is used to perform a toxicological risk assessment, which includes research into existing data about each chemical and composition's potential hazards with regards to specific biological endpoints. Again, areas where notable differences are found should be flagged for further investigation. Some typical items of interest for chemical property characterization are percent extractables in ethanol, methylene chloride, and saline. Comparative chemistry of the extractables in hexane and saline using GCMS and FTIR. Crystallinity of the reinforcing fillers using X-ray diffraction. Glass transition temperature using DSC. molecular weight of the extractables using HPLC, GC, and GCMS, surface characterization using a scanning electron microscope, thermal stability using TGA, and an IR spectrum. These all also have ASTM standards, ISO standards, and USP standards that can be used as guidelines. Next slide, please. In the absence of reliable literature on the effects of the chemicals present in a material on these biological endpoints, testing the material itself may be necessary to understand its biological performance. Testing at the material level does not reduce or replace the need for end-use specific testing. However, it helps customers determine the viability of using a particular material in their application. Areas where notable differences are found between the generic device and established device's materials should be flagged for further investigation. Typical key items of interest for biological property characterization are percent extractables in ethanol, methylene chloride, and saline, comparative chemistry of these extractables again in hexane and saline using GCMS and FTIR, in vitro testing for cytotoxicity, a study for systemic toxicity, a study for acute intracutaneous reactivity, 90-day implantation test, in vivo toxicity, a rabbit pyrogen study, delayed contact sensitization study, and in vivo cytogenic damage. Multiple ASTM test methods as standards and USP standards exist to cover these characterizations, either generically or for specific end uses. Next slide, please. Our final section, additional areas of consideration. Next slide. Some additional areas of consideration when choosing and characterizing a silicone material for use in drug delivery applications are specifications and specification justifications of the selected material, manufacturing process and process controls for the selected material, control of materials throughout the supply chain, control of critical manufacturing steps, and of the selected materials intermediates, process validation of the selected material, the manufacturing process development, the stability summary and conclusions of the selected material, active drug master file or master access files filed and updated with the FDA. Next slide, please. And we can move to the final slide. One more, please, for time. All right, to learn more about silicone technology and the products NuSil offers, please visit us at nusil.avantorsciences.com. Once again, I'm Matthew Kihara, and thank you for joining me on this presentation.
Thank you. Thank you, Matthew. And that concludes all the talks for session 1. And I would like to invite all the in-person speakers to the podium to start the panel discussion. And in addition, we also have Dr. Mohamed, Jafri, scientist, oral inhaled and nasal product development, Vectera, 13 farmer as an additional panelist. And we have Dr. Kinam Park and also Matthew online. So the first 10 minutes also we'll try to address specific questions you may have on any of the talks. And for the in-person audience, we have a microphone available. So if you have a question, please raise your hand and we'll pass a mic to you. And for online audience, please continue submitting your questions to the chat box. We will try to address them. Okay, so while the in-person audience warm up to prepare themselves, we do have a few questions from the online audience. So the first question to Dr. Lynd, the The question is, for your modeling work, did you use any software such as Lynd for solution of chemical kinetic equations?
Is There we go. So all the software that maybe that is on. Okay, all the software so we did use software, of course, but all of the software was written by us in C++. And we're happy to share it with you as well.
Thank you, Dr. Lynd. The second question is, I think as a general question, what do you think are the remaining challenges to really implement the model to really guide the actual manufacturing?
Well, I think right now, what we're doing with it is applying it to different polymer systems just to check the generality and robustness of it. But I think it already gives us a lot of what we need. It helped us. It was the machine that helped us establish a test for blockiness. And so I think that's really the key contribution there. And it's really a question of how that catches on. And then also, the next phase I would say is really more of an experimental study where we've got this method of measuring, a very robust method of measuring blockiness now. Now we can look at to see how does that affect properties. Even if it turns out that it doesn't affect them, you still have to understand the blockiness in order to do that.
Thank you. The next question. Oh,
hold on. I think.
Yes, please go ahead. Sorry.
Good morning. I'm Scott McNeil from University of Basel. Thanks to all the speakers this morning for giving excellent presentations, especially on the various characterization methods and how much we've improved over the last 20 years. And more specifically, you gave great examples of how these excipients are, in fact, very complex to characterize. So as these new methods become online and we validate them, the list of parameters and tests that regulators could ask expands and expands and expands. And so it occurred to me, I wondered if the panel and those online have any comments on what would happen if we actually went back to the reference listed drug and examined it based on all these new factors. Because at some point, we certainly want to not just make a list of physical chemical parameters, but then map those to CQA's critical quality attributes where they do influence product quality. So my question is, just in retrospect, how does that compare to the reference listed drug, and what measures are we taking to truly map those against CQ?
Actually, in my perspective, is a reference list drug is when they developed. There's a lot of research for the quality perspective. Also, a lot of research for the clinical perspective. When we just developed in the generic product, and in agency-wide, when we evaluate for the generic drug product, we just missing the one information for the clinical test. And so the best way we can approach how the same as RLD, that is our best. Actually, the Dr. Park's talk, he just asked for why is there so many different complicating the PLGA types, why is the RLD is developed. I think that is, in my opinion, they just in the developing for their own purpose based on their clinical study and what they need. And currently in the regulatory perspective for the evaluation of the generic product, we have to focus on the exactly same as RLD. But there is another big challenge because in polymeric material, there is no identical polymeric materials. That's the big challenge. And so now we can approach for the kind of the concept for the statistical identical concept, which is in the why we describe the loss of test, need to figure out why it's the same as in the RLD. That's in my perspective. You can.
Hi, Mark and Luke. I just want to be cautious there that we are not advocating necessarily for clinical testing of materials for bioequivalence for similarity, because I think within itself, the clinical testing, there's a lot of inherent variability, and an in vitro test that could produce results that mimic a clinical environment in a consistent manner is so much better than actually testing it in people. People do, and there's some logistical issues about sticking an implant into a person for that just a reason of testing it. I don't think people, well, some people might want to volunteer if they get paid enough, but I don't think that's a very practical thing to do. Thank you.
So one additional comment I have on that question is you also have to think about if you were going to start applying a bunch of analytical methods to the RLD to characterize and then compare with excipients, raw unprocessed excipients, is you also have to concern yourself with the effect of the manufacturing and sterilization of the drug product, if it's a terminally sterilized product, on the chemistry of the material. So for example, if you take a polymeric implant, right, and it's terminally sterilized, you have very likely altered the molecular weight distribution of the polymer through that terminal sterilization process. You know, at least with ethylene vinyl acetate, you've cross-linked the polymer. So you actually can't now take it and do GPC analysis and characterize it and compare it against a like an unprocessed polymer. So that's another, I think, complexity in that.
Yeah.
Hello.
Yes, baby.
Okay, yeah. I just wanted to follow up on the second question for Dr. Lynd, which is that it seems like there's some pretty good proportionality between the blockiness, the overall composition, and the extent of reaction. But of course, the extent of reaction is also dictating the molecular weight. So how, what kind of hope do we have for controlling specific molecular weights and blockiness and combination instead of kind of letting it be random, I guess?
Yeah. So Billy, thanks for taking us back to that question. There was, because answering that question was another thing I thought I should add is so that model really, right, is the machine that helped us generate a tool for blockiness. But the question I think was about how can this help manufacturing? The answer is, well, we've got a model of all the chemistry now that's properly parameterized. So that model could really help take a lot of the mystery out of the synthesis of PLGA or provide tools for controlling processes that create PLGA. And so within that context, we could look at all of these aspects separately, molecular weight distribution, and there were, we could target a certain blockiness as well, or look at a material that, that's already out there in the wild and say, well, in the context of this batch reactor, you would run the process for 17.5 hours to make the same thing in that reference.
My name is Jarni. I'm coming from Sandoz company. So coming from industry, I really appreciate all the examples you presented on the mock deficiency. But if you please, I would like to raise a deficiency to the deficiency answers that you provided. So in industry, what is important, these examples really present us an idea how to shape our development strategy and everything. And the list of tests that needs to be done is quite clear, quite extensive. But what we are usually missing is also, okay, we will do the tests, we will generate the data. First, what to do with the data, statistical approaches. Sometimes it is clear from the data because the data dictates whether it's a numeric or spectral data, whatever. But also the second point is, what is then the acceptance criteria? What is deemed comparable? This is also a question because it is important that we set the right target so that also the development is then successful.
All right. There is in the two kind of the approach for the making the sameness, the demonstration of the sameness. Most of the demonstration of the sameness issue is the one-time demonstration study. And so there is what kind of the exactly, well, lots of form just ask us for the, hey, what is the exactly number for the comparing? We cannot answer it, but you can from the lots of the common research and some guideline from the FDA, you can get into some idea how you can demonstrate of the sameness. And then another perspective is the quality control. That is different routine control. And so into that kind of things, you can get into some of the specific number for the acceptance criteria very detailedly. But in that case, the most of the demonstration sameness, if we has the very critical item, we can just ask you to include to the routine control. But other than that, you just know one time your manufacturing capacity can develop or developed for the apples to apple for the your product and R&D product. Is that enough?
Thank you.
Yeah, if I could chime in here a bit, I know I'm a moderator. So in terms of acceptance criteria, really for the same material, it can be very product specific. So how to establish sameness, that was one of the very original questions which promoted us 10 years ago to really start all these research projects to better understand the excipients and different formulations. And I think that goes back to also the earlier question with all those expanded methods, knowledge, where we are heading towards. So I think one thing to be clear is we need to get to the bottom of certain aspects to better understand it, to be able to make the right decision. So certain things we're evaluating does not necessarily mean we're gonna rigorously require those, but that's our process to really understand the role and the impact. If our research or our study shows maybe it's something good to know, but not like it's not must to know, then we will, not set that as part of our standard. So I think that's really one thing we should keep in mind. And then, for generally speaking, then for parenteral products, the sameness excipient, there are two stages. One stage is filing, and then is the final approval. So filing is really, we're trying to keep it as the minimum bar, right? Then for PLG, the reason we ask for more comparative is the recognition of the changing to the materials and depending on the manufacturing. So it's really, we feel it's really for the applicant's benefit to know what's your endpoint, what's the target. Although we know now, if you know the target, you may still don't know where to start. That's a separate story. But then, so we keep certain like molecular weight distribution, LTG ratio, structure, those are as basics. But then that being said, when it comes to molecular weight, for instance, for some products, we know if you have more than 20 kD difference, you may not see a difference in vitro release or clinical. But some products within 5 to 10, you see a big difference. So that's where it's up to every applicant to really understand your product and provide data to us to justify. Although on face value, you see some differences, why it doesn't matter. And then that would be, as a totality of evidence. You could leverage from your comparative characterization, you could leverage from your clinical studies. So those are some feedback.
So in the context of the reference listed drugs, even if you have a full characterization of the polymers, well-defined characterization of all the parameters, there is always this challenge of the innovator drift, the R&D drift. In terms of, say, the process of making the product, there could be an inherent variability even with the innovative product. In those types of instances, because of the RLD drift, it could be very challenging to get the sameness or the equivalency establishing. So how do you, what is the agency's, thought process in that? Is there anything that we can provide in terms of our understanding to make these generic drugs to be more feasible? Any comments on that?
Yeah, thank you for bringing up. I think that is a more general, like broad question, which we could have more in-depth discussion during the small roundtable discussion. But generally, I think, again, our strategy is really science-oriented. That's the whole reason we are doing this much research and trying to connect with the generic applicants during development. So I feel it's a two-way process. We do what we see as priority to provide as much understanding as we can, but then it's really critical get feedback from you guys to really help us to calibrate what we think you are struggling with are truly your struggle. So I think that's where this kind of workshop or other, like for specific program you are having, then the pre-enda, for instance, meeting requests or controlled correspondence, those are the right channels for you to really engage with us early during the development. If you have any ambiguity on your direction, but you have done some homework, you want to get feedback, that's a good way. Okay, I think we can.
Maybe I can give them the two cents for the comment. As I mentioned before, that is very challenging for the complex polymeric excipients, and also is some of the drug product, polymeric drug product, because as I mentioned before, it's a polymer itself. We cannot give them the identical analysis. And so we just giving to the lot of information for them some of the critical tests. And so you can get them some idea for them to providing all the sameness demonstration within the today and tomorrow's talks in the highlighted test, and then figure out for them your product is same as RLD. And then that's maybe very helpful to evaluate your product is in the approvable or not. Thank you.
Okay. And I have one more question here. So for assessing product sameness and also assessing manufacturing consistency, I wonder, is it useful to have an analytical technology that doesn't involve any sample preparation, but on the other hand, it doesn't measure a recognized parameter currently being measured? Is it useful to have such a new technique without any sample preparation? So that's my question.
Can you ask one more time?
Yeah, the question is, in order to assess product sameness and to assess manufacturing consistency, is it useful to have an assay or analytical technique that doesn't involve any sample preparation? Because I heard in earlier presentation, sample preparation can affect the assay result. So a technique doesn't involve any sample preparation. Will that provide additional value?
It's a excipient assay or API assay?
It's the product assay. Say you have a finished product that can measure the finished product, say without opening the vial. Will that be useful?
I quite don't understand your question, but if it's due to for my understanding, there is in the demonstration of sameness of the excipient itself has a one-time demonstration, and then for the drug product sameness, we need to I didn't especially in the separate within the one-time test versus a routine control, but API assay is a sort of the routine control test. And so that is in the analytical method, and how the acceptance criteria for the API assay is you can submit through the ANDA, or maybe in the freely asking to agency, we can just give them some of the hint. So that's, I don't know if my answer is what you want, but that's my best.
Excuse me. So, thank you very much for all of the presentations. I learned a lot this morning, and I have a couple questions for Kinam online, Dr. Park. And so as he was discussing the characterization of the star polymer and also trying to separate out different types of polymers within a blend, the first question is, some of our work has strongly suggested that there are linear chains that are present in these star polymers. And so the first question is, have you looked at ways to determine, and measuring, for example, just simple acid number of the polymer, because the linear chains will have a carboxylic acid on the end. So have you evaluated these different polymers and seen differences in the amount of linear chains? And the reason that I asked that is that the linear chains would be the ones that can form a salt with the peptide that is thought to be a precursor for the acylation reaction. I'll ask the second question after he answers.
Okay, I think the organizer does not allow me to start my video, so I will just talk if you can hear me. That's a good question. Linear PLJ is always, well, here you go. Linear PLJ exists, but the amount, according to our analysis, is only less than few percent, like less than 5% maximum. And the reason is that if the PLJ molecular weight is so low, then they may not have enough grafting into glucose, first of all. But the total amount is no more than few percent, 5% maximum, I guess. So the linear PLJ may exist, but it's a very minor factor. So I think majority of them are glucose PLJ, but the branching number may be different, but majority of them are branched star polymers.
Thank you. The second question is, you were talking about separating different types of polymers with their solubilities, different lactide glycolide ratios, and so on. And my question is, I would imagine that the solubility of the polymer not only depends on how the ratio of this, its molecular weight, et cetera, but also the molecular configuration of the solid. So do you see that? I mean, if you have more interchain entanglements, I mean, you're probably getting a pass in the sense for a lot of lower molecular weight compositions. This may be less important, but I'm just wondering, have you considered the potential effects on solubility based on the solid state structure? I mean, in a similar manner, you see differences with different crystals of different drugs on the solubility, but of course, these are amorphous polymers. But if you see, if they've been recrystallized differently, if they have different blends that could have different entanglements, et cetera, would that affect their solubility?
Right. That's the basis of the concept of semi-solvent. Depending on the LG ratio, different semi-solvent dissolve them. And here, when we tested all this semi-solvent, we had the molecular weight less than 100,000. So as you said, if the molecular weight is like a million, couple of millions, then it may behave differently. But eventually, their dissolution kinetics may be different, but eventually they're going to dissolve. So the importance of blockiness is that depending on semi-solvent, semi-solvent cannot diffuse into the crystal region of the glucolide. So that's why blockiness is important. But the configuration is a large molecular weight, like 1 million Daltons. They may take a longer time to dissolve, but eventually they will dissolve. So I think all our assay was done with a molecular weight less than 100,000 because most of the PLJ used in commercial product are less than around 100,000.
Thank you, Dr. Park. And Jafri, if you'd like to chime in back.
Hello. I just want to respond to your question earlier on the analytical method without any sample preparation. So actually, yeah, some cases, it is preferable. So it is, I think, depends on the purpose of the analytical characterization. Some of them, if the sample preparation will affect the result, if you can have a method that you can really characterize that perspective without sample preparation, that is very favorable. Yeah, that's just want to.
Thank you. And for the last remaining minutes, we will try to address a couple online questions. So one question for Dr. Haley is, can the extruded EVA samples be used directly to do DMA analysis, or is there any sample preparation needed?
So let me repeat the question so make sure I caught that right. So the question is, can the extruded EVA samples be used to do DMA analysis, or is there any directly? I mean, yeah, you can put a cylindrical rod, for example, into a test fixture in a DMA and do sort of like a tensile oscillation with it. I don't see why you couldn't do that. The one thing is, if you're characterizing a multi-layer sample, I would, most of the cross-section of that sample is really going to be the core layer. So I doubt you would pick up very much, although you might see, some differences in the transition temperatures of the skin layer, but most of it would be the core. But yeah, I think you could directly load a sample like that into a DMA.
And then another question, I guess, is asking if you have the same V content, but you may still have different melting index, and why may be the reason?
Well, so I mean, they're independent variables, right? So the melt index is really a function of, to a first approximation, it's a measurement of molecular weight, which is really going to be a function of the relative rates of propagation and termination and chain transfer and all the various things happening. Whereas the vinyl acetate content is really just how much vinyl acetate is introduced into the reaction mixture. So they're independent, right? There are certain cases where incorporating a lot of vinyl acetate into the polymer will start to suppress, because vinyl acetate's a chain transfer agent itself, will suppress molecular weight. But you can have a 28% vinyl acetate copolymer with a melt index of 3 or a melt index of 800. It doesn't really, they're independent parameters.
Thank you. And the last question we have from online audience is for Matthew. So can you share any specific parameters you think is the most challenging for characterizing silicon elastomer polymer?
Yes, for characterizing the silicon polymers, I would say the most challenging is getting to what the chemical compositions are. Because with these silicones, you're dealing with reactive chemistries. They're thermoset polymers. They're not they're not your typical plastics that you can melt. You're actually running chemical reactions in the presence of the APIs. So any different reactive groups or functional groups, even if non-reactive, may play a critical part in how the device actually performs. So the silicon formulations do get quite complicated when it comes to API elution due to this.
And then a related question is, given the material, the silicon elastomer in finished product are generally cross-linked, so do you have any recommendation on the most effective analytical methods to characterize or trying to compare the composition or properties?
Yeah, we can follow up with more details. Some of those are listed within the presentation. Physical properties are a good guideline, but they don't get to the chemical portion. To really find out what chemicals are in there, it may require some like IPC, and then IR spectroscopy and many other analytical methods to get to the various aspects in there. Because these silicone formulations, it's not just polymer. There's multiple fillers, multiple additives in there as well. And a lot of these will use different cure chemistries. Some of those catalysts stay in there. Some of them are removed. Some of them, the functional groups are gone by the time it's in the cured state. some of these silicones, the functional groups remain.
Thank you. And to keep everything on track, we will have one last in-person question.
Yeah, thank you. So my question is related to PLGA grade. Excuse me. When we are developing generic long-acting injectable product, and if we use a PLGA which has slightly different attributes compared to what RLD has used, especially if we talk about the viscosity. So if we show the same in vitro and in vivo parameters and sameness, can we use the PLGA which has the slightly different attributes compared to RLDs?
Again, it's in the I cannot answer directly. It's in the how much in the slightly difference is difference. And so if we can see that, and then it looks like it's a reasonable difference, and then we may be in the accepted, but otherwise, why is in the yours properties is different with RLD's properties, such as something is from some manufacturing properties, manufacturing process parameters difference, or some other design of the development is different, and we cannot tell exactly because as I mentioned before, this is the polymeric material sameness is not exactly identical, and so we cannot compare with apple and apple, and so we can just see it in the oral actual data, and then we can evaluate. Thank you.
Okay, thank you.
The very last question, Eric Witschow, AMW. Thanks a lot for this interesting presentation, and it was clear how fascinating the field of polymers are. But I think we hopefully all agree that all the parameters you presented are not really related to safety of the product. It's all performance related, and performance normally means release test and pharmacokinetic. The reason why we spend now so much effort in improving the analytical method isn't, and that's the question, is it because we don't trust our in vitro release methods, and there must be maybe more effort in improving the release methods instead of analyzing the polymer itself?
Well, I trust you guys first. But today's we just shared in our knowledge is, I don't know, the yours is the same as in the other yours. And so we just roughly generalize, we need our expectation, we need to require for this kind of the test and test method can support to the sameness, and then we can evaluate for the yours is same as RLD. We not evaluate for the yours and other yours.
Can I add one
Sorry.
thing here? Can I add
Yeah,
one?
I know it. Oh, sorry, Dr. Park.
Just one. Using in vitro release only as a comparison is dangerous because in vitro release can be done in a variety of different conditions. You can make the same. That alone is not enough to approve as a generic. That's why you have to analyze ingredient to make sure that they're similar, at least similar, if not exactly the same. So that when you have a same in vitro release profile, you can reasonably expect that it can have a same PK profile in human. This is why you need to analyze the ingredient. Otherwise, simply showing in vitro release profile the same is not enough. You just cannot guarantee that they will have the same property in vivo.
Thank you, Dr. Park, and thank you everybody for your attention. And we have to end session 1, and we will have more discussion in person later. And now I'll pass the podium to the moderator, Dr. Bing Ching, for the second part of the workshop. Thank you very much.